Dear students, after studying this module, you shall be able to know about the structure and functions of biotin, coenzyme A, TPP, that is thiamine, pyrophosphate, and B12. You shall also learn about the mechanism of function of these coenzymes and analyze roles of thiamine phosphate as a coenzyme. Now students, let us introduce to you the topic of coenzymes. What are coenzymes? Students, coenzymes are non-protein molecules that function as essential part of enzymes. Coenzymes often serve as special reagents needed for reactions that would be difficult or even impossible using only simple acid-base catalysis. In many instances, they also serve as carriers, alternating catalysts that accept and donate chemical groups, hydrogen atoms, or electrons. They are often complex organic structures that cannot be synthesized by some organisms, mammals in particular. Now students, I would like to state that the water-soluble vitamins, those usually referred to as vitamin B complexes, are metabolic precursors of a number of coenzymes. That is why vitamins are so important in our metabolism and in general for metabolism. Coenzymes are considered under different groups depending on types of reactions that they carry out. In this module, we will consider those group of compounds which are derivatives of vitamins that, while in the active site of enzyme, alter the structure of a substrate in such a way that permits it to react more readily, or we can call them group transferring coenzymes. And we shall be studying in detail in this module, students, biotin, coenzyme A, TPP, that is thiamine pyrophosphate, and B12. Now let us begin our discussion of coenzymes with the discussion on biotin. First, emphasizing on its structure. Biotin is an imidazole ring fused with thiophene ring with a valeryl side chain. As you can see here, the structure of biotin is depicted in this figure. Biotin is widely distributed in food as biocytin, which is biotin bound to amino group of lysine. Now here you can see the enzyme bound biotin and its structure. That is the structure of biocytin. After having studied the structure briefly, let us now come to the function of biotin. This water-soluble B group vitamin participates in transfer of carboxylic group. Two important enzymes carrying out carboxylation are, first is pyruvate carboxylase and the second one is acetyl CoA carboxylase. Now students, what is the mechanism of carboxylation? A holocarboxylase synthetase catalyzes the attachment of biotin to eta amino group of lysine residue of carboxylase enzyme to form biocytin in an ATP dependent reaction. Now students, this reaction with the help of ATP is basically shown to you here in this figure. As you can see that pyruvate is converted into oxaloacetate. On the other hand, carboxylation enzyme is doing this and along with the biotin enzyme with the consumption of ATP. Biotin as a part of carboxylase enzyme then catalyzes the transfer of carbon dioxide to the substrate. It first attaches to activated carbon dioxide in the form of bicarbonate and anion, that is HCO3- to form carboxylated biotin. This then transfer carboxylate to the substrate. Enzyme bound biotin acts as a carrier for carbon dioxide in pyruvate carboxylase reaction. The biotin dependent enzymes require ATP, which is converted to ADP during the reaction. That is, the triphosphate, adenosine triphosphate, is converted into adenosine diphosphate during the course of the reaction. Pyruvate carboxylase is a tetrameric protein carrying 4 molecules of biotin, each bound covalently through an amide bond involving eta amino group of lysine residue. Pyruvate carboxylase operates by a two-step mechanism which is shown to you here clearly in this figure. As you can see, the two steps are involved here in this reaction. Also, you can see here that enzyme-bound biotin acts as a carrier for carbon dioxide in pyruvate carboxylase reaction, starting with an ATP-dependent carboxylation of cofactor to give N-carboxy-biotin. This activated derivative then transfers the carboxyl group to the pyruvate. After having discussed, students, the structure and the function of biotin and the reactions involving biotin, let us now divert our attention to another coenzyme, which is coenzyme A. Coenzyme A is an acyl activating enzyme derived from vitamin pantothenic acid. Pantothenic acid is a pentoic acid and beta-alanine joined together in an amide linkage. Now here you can see that pentoic acid is actually attached to the beta-alanine to give you pantothenic acid, which functions as coenzyme A. Coenzyme A can be divided into two components, adenosine 3,5-diphosphate and pantothene, which is formed by the combination of pantothenic acid and beta-mercaptoethylamine. Now, as you can see here, clearly the structure of coenzyme A here in the figure. Let us now study the structure. The SH group of thioethanol amine moiety is an active group acting as a carrier, and coenzyme A is abbreviated as COA, in which remains as a small letter, or COA-SH. The rest of the molecule provides enzyme binding site. In acylated derivatives such as acetyl coenzyme A, the acyl group is linked to thiol group to form an energy-rich thioester. For example, you can see here the formation of acetyl CoA with coenzyme A and the acetyl group. Coenzyme A reacts with many compounds to form important derivatives such as acetyl CoA, succinyl CoA, HMG CoA and fatty acyl CoA. Acetyl coenzyme A and succinyl coenzyme A are important intermediates at crossroads of many metabolic pathways. Acyl CoA is formed as an intermediate both in fatty acid biosynthesis as well as oxidation reactions. HMG coenzyme A is an important intermediate involved in the ketogenesis as well as cholesterol biosynthesis, and we designate the acylated forms of coenzyme A as, like you can see here, the acyl coenzyme A is represented by the R alkyl group and rest the same as acetyl CoA, and unacylated is represented as COASH, as described to you earlier, students. While coenzyme A was discovered as the acetylation coenzyme, it has far more general functions. It is required in the form of acetyl coenzyme A to catalyze the synthesis of citrate in the citric acid cycle, or the tricarboxylic acid cycle, or the TCA cycle as we popularly know it. This coenzyme is essential to the beta oxidation of fatty acids and carries propionyl and other acyl groups in a great variety of other metabolic reactions. The acyl group such as acetyl or acetoacetyl group is conjugated or attached to the coenzyme A through a thioester linkage to the beta-mercaptoethylamine moiety. Acyl groups are covalently linked to thiol group, forming thioesters. Because of their relatively high standards of free energies of hydrolysis, thioesters have high acyl group transfer potential and can donate their acyl group of variety of acceptor molecule. The acyl group attached to coenzyme A may thus be thought to be as activated for group transfer. Now students, I have described to you biotin and coenzyme A. Let us now focus on another coenzyme, that is coenzyme B12. Coenzyme B12 is the cofactor with the vitamin B12. This vitamin is unique amongst all the vitamins in that it not only contains complex organic molecule, is having an essential trace element, cobalt. The structure of vitamin B12 is seen here in this particular figure. As you can see, it contains complex organic molecules and also contains a trace element, cobalt, in its 2+ oxidation state. The tetrapyrrole ring system, called a Corin ring, is similar to porphyrin ring of heme-based compounds. Famous example is hemoglobin, which is a part of blood. The cobalt is also linked to heterocyclic base 5,6-dimethyl 1-benzimidazole, that is DMB ribonucleotide. This is clearly seen in the figure. In the vitamin, as it is isolated, 6th coordination position of cobalt is occupied by cyanide ion, but this ion is introduced during isolation. The vitamin, as it exists in tissues, contains either water or a hydroxy moiety at this site because of presence of cobalt and many amide nitrogens. B12 compounds are called cobamides or cobalamins. B12 derivatives are also named in terms of groups occupying the 6th coordination position in the entire molecule. Thus, the vitamins as isolated is cyanocobalamin and intracellular forms are aquacobalamin and hydroxocobalamin. What is a coenzyme form of B12? Let us now divert our attention to this topic. Two active forms of vitamin B12 are involved in the metabolism. These are methylcobalamin and 5-deoxyadenosine cobalamin, also called cobamide coenzyme. Only two reactions use this vitamin B12 as coenzymes. First is the conversion of homocysteine to methionine. Methylcobalamin is the coenzyme in this particular reaction. Second reaction using this coenzyme is the conversion of methyl malonyl CoA to succinyl CoA. CoA is the coenzyme A, and 5-deoxyadenosine cobalamin is used as a coenzyme in this reaction. Now you can see this reaction clearly in this figure as shown to you here. Here you can see that methyl B12 is the coenzyme in the synthesis of methionine from homocysteine as the first example that I took, first example of the conversion. Function of 5-deoxyadenosine cobalamin involves isomerizations involving exchange of carbon-bound hydrogen with another carbon-bound functional groups. As shown here, methyl malonyl CoA mutase catalyzes this reaction. As you can see here, the catalysis of the conversion of the L-methyl malonyl CoA to succinyl CoA. Let us now focus our attention on the study of thiamine pyrophosphate. Let us now introduce you to the structure of this molecule. Thiamine was the first of B vitamins to be identified. It is also called as vitamin B1. The vitamin is structurally complex and made up of 2,5-dimethyl-6-aminopyridine joined to 4-methyl-5-hydroxyl-thiazole by a methylene linkage. But its conversion to the coenzyme form, thiamine pyrophosphate or TPP, involves simply and ATP-dependent pyrophosphorylation. Thiazole ring of the TPP is the functional part of this coenzyme. This can be seen here clearly in this figure. Dietary thiamine after absorption is converted to thiamine pyrophosphate by thiamine kinase in most tissues of our body using adenosine triphosphate, that is using the energy of the body. Role of thiamine phosphate as a coenzyme. Now let us study this role. Thiamine pyrophosphate participates as a coenzyme in reactions. First one is the oxidative decarboxylation of pyruvate, a ketoglutarate and branched chain amino acids. Second one is a keto acid dehydrogenase reaction of branched amino acids. Third is a trans-ketolase reaction of HMP stunt pathway. Thiamine pyrophosphate is the coenzyme for all decarboxylations of alpha keto acids. The mechanism shown to you in the figure for pyruvate decarboxylation is involved in all of these reactions. Note students that TPP contains two heterocyclic rings, a substituted pyrimidine and a thiazole ring. Recent NMR studies have shown that both rings participate in the formation of a reactive carbanion at C2 of the thiazole ring, that is the carbon atom between the nitrogen and the sulfur. As shown in the diagram drawn here, a glutamate carboxyl group in the enzyme attracts a proton linked to the N1 of the pyrimidine, which in turn increases the basicity of the amino group, thus facilitating the deprotonation of C2 of the thiazole ring. Students, note that the carbon forms a carbanion in the step 1 as shown to you in the figure, which in turn can attack the carbonyl carbon of alpha keto acids such as pyruvate, giving an additional compound. The addition compound then undergoes non-oxidative decarboxylation in the next step, which is the third step, with the thiazole ring acting as a stabilized enamine. Protonation gives a hydroxyl TPP as is shown here in the figure. Thiamine pyrophosphate in the pyruvate dehydrogenase reaction. TPP, or that is thiamine pyrophosphate, is the coenzyme for the pyruvate dehydrogenase reaction and other non-oxidative decarboxylations of alpha keto acids. The key reaction is the attack by the carbanion of TPP on the carbonyl carbon of pyruvate and is followed by non-oxidative decarboxylation of the coenzyme bound pyruvate. The electron pair remains with the ring nitrogen. In the step 4, the 2 carbon fragment bound to TPP extracts a proton from the pyruvate decarboxylase, generating a hydroxythyl group or the hydroxyl group. This fragments remains at the aldehyde oxidation level. Now students, after having studied so many coenzymes like biotin, TPP, B12, let us summarize this particular module, in which we have taught you that 4 enzymes which are involved in the transfer of groups are vitamin B12, biotin, TPP and coenzyme A. Biotin participates in the transfer of carboxylic groups. Thiamine pyrophosphate or thiamine TDP is the coenzyme responsible for the transfer of aldehyde and glyoxyl group and is derived from vitamin B by phosphorylation. Coenzyme A is derived from vitamin pantothenic acid. This is abbreviated as CoA. It is also called as acetylation coenzyme. Coenzyme B12 is the cofactor form of vitamin B12 and is involved in the isomerization reactions as well as the methyl group transfer reactions. So students, I hope this module gives an insight into the various cofactors or coenzymes which are involved in the metabolism.